And what happens here is that recurrent implantation failure is a complex problem with a wide variety of etiologies and mechanisms, as well as treatment options. The recommendations for women with RIF vary depending on the source of their problem. Perhaps the best and yet most complex answer is personalized medicine, a personal approach to each patient depending on her unique set of characteristics. So it's a complex situation, and no single biomarker or factor can really address this complex situation. So I just want to take you through one assay that is being widely used currently in the IVF clinics, and that is endometrial receptivity assay. And what it does, why it is widely used? Because it uses endometrial biopsy in a mock cycle to establish the best day to transfer the embryo. The ERA, which is the endometrial receptivity assay, will determine if the endometrium is receptive or not at the time of sampling by analyzing 248 genes involved in endometrial receptivity. ERA testing can find your personalized window of implantation in case of displacement and will allow a personalized embryo transfer that is recommend more or less progesterone prior to embryo transfer. Another benefit that people are talking about is that local endometrial injury improves the pregnancy rates in subsequent frozen embryo transfer. So let's talk about uterine NK cells. We and others have published a lot on this subject matter. And basically this slide shows during menstrual cycle when, or even during pregnancy, when these cells reach the peak. And basically in menstrual cycle what happens is that during secretory phase they reach the peak, and if the pregnancy does not take place they are sloughed off from the tissue, and if pregnancy takes place then these cells keep increasing and reach the peak during early part of second week, and then they taper off after that. And they are very important in terms of communicating with invading trophoblasts, and they are also very important, which I will discuss in the next slide, that they also support angiogenesis and immune tolerance. So we wrote an article a few years back suggesting that in normal pregnancy, conditions like hypoxia or factors like TGF-beta or even epigenetics mechanism can convert these cells into regulatory NK cells, and they produce factors like VEGF, PLGF, interferon gamma inducible protein IP10, IL-8, IL-10, GM-CSF, and some of these factors support angiogenesis, also induce immune tolerance, for example, cytokine like IL-10, which is anti-inflammatory, and that contributes to normal pregnancy outcome. On the other hand, we have published, and when we published it, it was not well accepted, but now others have published the same thing, so I think this is quite valid. What I'm going to talk to you about is that intrauterine infections, whether bacterial or viral infections, or even progesterone withdrawal, can turn these cells into inflammatory cells, and they start producing cytokines like TNF-alpha or express molecules like NKG2D, and they acquire cytotoxic phenotype and blunt immune tolerance and angiogenesis, and may lead to adverse pregnancy outcomes such as fetal loss, preterm birth, and even preeclampsia. So these cells are quite detrimental in the context of viral or bacterial infections. On the other hand, deficiency in these cells can also help the propagation of T helper 17 T cells, which can contribute to further inflammation and blunt immune tolerance and angiogenesis. So these cells are quite important, and by virtue of regulatory in the nature and the functional phenotype, and they also represent the predominant immune cell type at the mental-fetal interface. So they play a major role in a normal pregnancy outcome. Many people, many labs have talked about the significance of peripheral blood as well as uterine NK cells, and number of these systemic review and meta-analysis studies have told that have failed to show any relationship between peripheral blood NK cell number or activity or uterine NK cell density as a predictive marker for pregnancy outcomes. Many of these authors further concluded that the role of NK cell assessment remained unclear, and that more studies were needed before it could be determined who might benefit from either screening or immunotherapy. So, what we are really talking about here is that there must be some other cell types that are involved in maintaining immune tolerance or providing help to the fetal growth and development. So we just talked about NK cells, and there's another cell type, immune cell type, that has become quite important at the maternal-fetal interface are the regulatory T cells. They are specialized regulatory T cells, and what we think now that they are essential for protecting the fetus from immune rejection. In mouse studies, anti-regulatory T cell antibodies administered shortly after mating causes complete implantation failure. Immune tolerance against alloantigen specific effector CD4 cells generated in response to fetal antigens, and they produce cytokines, anti-inflammatory cytokines such as IL-10 and TGF-beta, and promote anti-inflammatory tolerogenic phenotype in alternatively activated macrophages and tolerogenic dendritic cells through TGF-beta, IL-10 and CTLA4 mediated mechanism. And it has been suggested that their depletion before implantation, as I mentioned earlier, leads to complete implantation failure. We still do not know what is their significance during the active phase of pregnancy post-implantation. The experiments are still not clear on that, so we still need to do more work on delineating the exact role of immune cells in pregnancy. Immune cells means regulatory T cells in pregnancy. So the clinical questions that we are trying to discuss here: can we use peripheral blood, NK cells and Tregs to predict pregnancy outcome? Can we use endometrial NK cells and Tregs to predict pregnancy outcome? Can we change peripheral blood NK cell phenotype or function during an frozen embryo transfer cycle? If change occurs, can we use it to predict pregnancy success in IVF clinics? How does the endometrial receptivity correlate with immune cell density? So these are some of the clinical questions that we are trying to focus on. So this is the aim one that we set out in our studies, that states characterization of peripheral blood NK cells and Treg phenotypic status in patients undergoing assisted reproductive care. So our hypothesis was that in patients who conceive, a larger proportion of peripheral blood NK cells are CD56 bright, super bright, and CD16 negative. And patients who conceive have a higher proportion of Tregs in peripheral blood. So this was this constituted our aim 1, and the next slide tells you the study design, how we went about it. It's a prospective cohort study from October 2017 through present time. It's a powered analysis. It's based on hypothetical changes in the peripheral blood, CD56 positive, CD16 negative NK cell population in the pregnant versus non-pregnant state. We further hypothesized that pregnancy would result in a 25% increase in CD56 positive, CD16 negative NK cell concentration. It's power of 80% and an enrollment ratio of 1:5 and 1. Sample size 33 according to these power calculations, 13 in pregnant group and 20 in the non-pregnant group. And to account for cancer cycles and biochemical pregnancies, we enrolled 51 patients to the study. These are the inclusion criteria, inclusion and exclusion criteria. The inclusion criteria: phase 1, all patients undergoing FET cycle. Phase 2, euploid embryo FET cycles. Exclusion criteria: cancer cycles, no embryo transfer, spontaneous abortion, or biochemical pregnancies. These are the inclusion and exclusion criteria. Sample collection: we use 3 time points. So we collected in the proliferation follicular. These are blood samples that I'm talking about. Then we collected sample during secretory luteal phase, and that is to assess the endometrial thickness, and that's when we start progesterone treatment, 5-6 days. And after that, at the end of progesterone treatment, embryo transfer is performed, and then we wait for another sample that is to detect serum beta HCG to see whether pregnancy has taken place or not. So these are the time points that we have collected samples. So this is the follicular phase, and then progesterone stage, secretory phase, and then at the time of serum beta HCG collection. So these are the time points, and I'll take you through some of these samples, the data that we have collected. Now, so we went through sample size of N15, which is from the clinical pregnancy, and then we had 20 patients with negative pregnancy. And what we did here, we collected whole blood, isolated peripheral blood mononuclear cells, carried out flow cytometry for NK cells and regulatory T cells. We also did to assess the functional activity of NK cells, peripheral blood NK cells, we did endothelial tube formation assay, and I'll take you through those experiments. And finally, we also performed natural killer cell kill assay using K562 target cell line. So this is the experimental design for us. So the first set of data, and we did use number of experiments to characterize these parameters. And here we are looking at CD56 positive NK cells and different phases of sample collection, as I described to you in the previous slide, which is follicular phase, day of embryo transfer, day of serum beta HCG detection. So what we see here, and we have compared women who became pregnant and women who remain non-pregnant. So in those women, both the groups had similar NK cell population. There was no difference during the follicular phase. These numbers did not change either at the time of embryo transfer, which is after progesterone treatment. However, we saw high number of NK cells, CD56 positive NK cells, in a pregnant group at the time of beta HCG detection compared to non-pregnant group. So this suggests that the NK cell number does increase in pregnant women, and I'll show you later on that these NK cells in pregnant group are non-cytotoxic in their functional phenotype compared to NK cells from non-pregnant women. We did not see any changes in CD16 positive cells or CD56 positive NK CD16 negative cells. So it's basically a very subtle changes that we see in NK cell population in pregnant versus non-pregnant group at the time of beta HCG detection. So we also looked at in the same samples peripheral blood regulatory T cells, and again we are comparing samples from follicular phase, day of embryo transfer, and then beta HCG detection, and again we can see regulatory T cell number also increases in pregnant group at the time of beta HCG detection. Before this stage, we could not really detect any difference between NK cells, or in this case regulatory T cells, any difference between the cell numbers between pregnant group and non-pregnant group. The only time that we saw this difference was at the time of beta HCG detection. So the AM2 then talks about determination of peripheral blood NK cell functional characteristics in patients undergoing assisted reproductive care. So our hypothesis was that in women with failed implantation, cytotoxic NK cells have higher cell kill potential. So this is what we really wanted to go after, just to see. So again, it's an experimental design. We again took samples from 15 patients who had clinical pregnancy, and then 20 with negative pregnancy, collected whole blood, did phenotypic characterization. Then for functional characterization, we used endothelial tube formation assay, and then functional kill assay, or using K562 cell line as target cells. So this is the tube formation. The functional, the phenotypic characteristics I have already described to you. So this is the tube formation assay. We have published a lot on this thing, and what this is telling that we talk about spiral artery remodeling, and spiral artery remodeling basically involves endovascular crosstalk between trophoblast cells and endothelial cells. So we have mimicked that assay in vitro here. And basically what we are using here, UVEC endothelial cells and HTRA first trimester trophoblast cells, which are extracellular trophoblast cells. And when you do that, when we assay these cells here, these are endothelial cells, and by themselves, without trophoblast cells, they start forming three-dimensional structures. HTRA cells do not form any three-dimensional structures, and when we mix both of them together, you can see well-defined three-dimensional structures. And these are done in the presence of serum from pregnant women. And then the next three D, E, and F columns also suggest to you that we have used serum samples, and then serum samples plus NK cells from pregnant women and NK cells from non-pregnant women. So all D, E, F, these experiments are done in the presence of pregnancy serum from pregnant women, and with or without NK cells from pregnant women or non-pregnant women, the system does provide evidence that yes, three-dimensional structures are formed in the presence of serum from pregnant women. Now this is very interesting. G, H, and I columns, panels rather, show you that this is here you have UVEC cells and HTRA cells in the presence of non-pregnancy serum, and you do not really see heavy-duty tube formation as we saw here. And then, however, when we do the same experiments in the presence of pregnant NK cells, you can restore some of the formation of three-dimensional structures. However, in the presence of non-pregnancy NK cells and serum from non-pregnant women, the formation of three-dimensional structures is quite disrupted. And you can see here in this experiment, this data, what I just told you here, is depicted here. So you can see that non-pregnant serum plus non-pregnant NK cells do severely disrupt the formation of three-dimensional structures between endothelial cells and trophoblast cells. So this says that NK cells from non-pregnant women or serum from non-pregnant women in these IVF treatment groups do disrupt the endovascular crosstalk between endothelial cells and trophoblast cells. So these are the findings: no serum, no tubes. Then serum from preeclampsia patients, that is our control, which we have published, that serum from preeclampsia patients disrupts tube formation. Pregnant NK cells are not disruptive to tube formation. Non-pregnant NK cells, serum dependent. If you have serum from pregnant women, these cells are not effective in terms of disrupting tubes, whereas non-disruptive in the presence of pregnancy serum, but they are disruptive in the presence of non-pregnancy serum. So the combination of non-pregnancy serum and NK cells from non-pregnant women, they are quite potent in terms of disrupting tube formation. So this is one functional assay that we used. So the next question we ask: are pregnant NK cells less cytotoxic in general? And does pregnancy serum impact non-pregnant NK cell cytotoxicity? So this is the K562 cell kill assay. This is K562 cells are immortalized myelogenous leukemia cell line, originally isolated from a 53-year-old CML patient, and lack MSC class 1 molecule, and that makes them the target for NK cells. So this is just basically tells you that if the class 1 and the NCR on NK cells interact with each other, that leads to no killing, whereas that takes to the killing, whereas if there's no interaction between class 1 molecule and NCR on NK cells, that leads to killing. Whereas what I was trying to say before, that the class 1 and NCR interaction leads to no killing, whereas if you don't have class 1 molecule there, that leads to killing. So this is reverse of what cytotoxic T cells do. Cytotoxic T cells require interaction between class 1 and receptors on T cells to initiate cytotoxic activity. It's a reverse in NK cells. So that's why we use this K562 cells, which lack MSC class 1 molecule. So again, we have done these experiments in a number of ways in the presence of no serum or serum. So what we do in the presence of no serum, we take K562 and then PBMCs from pregnant women and PBMCs from non-pregnant women. So this is what you see here, that again, these PBMCs are collecting at the follicular phase or day of embryo transfer or day of serum beta HCG detection, and you can see in pregnant women at the time of beta HCG detection, the NK cell cytotoxicity much less than NK cells from non-pregnant women. So this basically tells us what we have been talking about, that the NK cells are non-pregnant in pregnant women compared to NK cells. So these are peripheral blood NK cells compared to women who did not get pregnant, who failed to get pregnant. So then we repeated these experiments in the presence of serum from pregnant women or from non-pregnant women, or non-pregnant PBMCs in the presence of serum. So this is the data again. So then you can see pregnant NK cells in pregnant serum, cytotoxic activity is quite reduced compared to non-pregnant NK cells in non-pregnant serum. And you can then come back and inhibit the cytotoxic activity of NK cells from non-pregnant women if you do the experiment in the presence of serum from pregnant women. So the serum from pregnant women contains some factors that are inhibiting the cytotoxic activity of peripheral blood NK cells. So that led us to characterize, we had published in the past that VEGF-C could inhibit cytotoxic activity of peripheral blood NK cells. So we compared production of these VEGF-A, VEGF-C in non-pregnant women versus non-pregnant, and we did not find any difference. Of course, pregnant group had HCG and non-pregnant group did not have HCG. That's nothing new, that's known, but this gave us an idea that each HCG inhibiting the cytotoxic activity of NK cells. So this led to new questions. Is HCG impacting peripheral blood NK cell cytotoxicity? So this is the experiment that we did here, where this basically we repeated the NK cell cytotoxicity assay on K562 cells, and you can see here that in patients, non-pregnant patients, where we these are NK cell cytotoxicity. These are cells from non-pregnant women NK cells, and you can see much higher cytotoxicity in these NK cells, whereas that could be inhibited by 10 international units of HCG, recombinant HCG, and you can see on the right hand side 3 different patients. We have done experiments, and you can see inhibition of cytotoxicity. Of course, it varies from patient to patient, but the common thread between these data are that yes, in all cases, the HCG could inhibit cytotoxic activity of peripheral blood NK cells from non-pregnant women. So this was one experiment. So the AM3 then we focused on characterization of endometrial NK cells and regulatory T cells, and basically in patients undergoing assisted reproductive care. So our hypothesis here was that in patients who do not conceive, a larger proportion of endometrial NK cells are CD56 positive, CD66, CD56 dim, CD16 positive, with a lower percentage of endometrial Tregs. So this is what we have done next, and this is the basically console diagram from the patient that we collected samples. So they are endometrial biopsy from 40 women, and we divided on clinical pregnancy and no pregnancy, 16 samples versus 17 samples. So we did immunohistochemical analysis. This is for endometrial NK cells, and you can see on the left hand side A sample is from a woman who did not get pregnant, and you can see the green represents CD16 positive cells, and red represents CD56 positive NK cells, and you can see in the non-pregnant the endometrium tissue from a non-pregnant woman does contain higher content of green signal, which is the CD16 positive red cells, whereas on the right hand side there is the endometrial tissue from a woman who went on to conceive, and you can see that we don't really have green signal in this tissue. And I'll show you the next slide that where we have done, we have seen so many patients, and the data are not very clear cut, but there's a trend towards a lack of CD16 positive cells in pregnant group, whereas this content increases in non-pregnant group. So this is what I was talking about. So we do not really find any difference in CD56 levels in endometrial tissue tissue in pregnant versus non-pregnant women. However, there is a trend for higher number of CD16 positive cells in endometrial tissue of non-pregnant women. So this is interesting. This requires a high number of screening of high number of samples, because we have only screened 17 samples here for non-pregnant and 16 samples for pregnant women. So we need to really screen very high number of samples to make some statistical statement here that yes, the difference in pregnant versus non-pregnant group as far as CD16 positive cells are concerned is statistically significant. And so we are continuing to do this work, and we would like to establish that yes, CD16 positive cells are much higher in the endometrial tissue of women who fail to get pregnant. But this is for regulatory T cells. So we basically did the same thing here, that you have on the left-hand side tissue from pregnant group, and then right hand side is tissue from non-pregnant group, and you can see here much higher number of regulatory T cells in pregnant group versus here versus the non-pregnant group, and this is the quantification of the data, and you can see this is statistically significant. There's a much lower content of regulatory T cells in the endometrial tissue of women who fail to get pregnant. So this data really is quite helpful, but it's laborious, and you would have to screen a huge number of endometrial tissue biopsy tissue to make some sense out of this data. So this is no difference in Treg density in a receptive cycle versus non-receptive cycle. So this is I was talking about the ERA assay. So we did not find any difference between receptive cycle or non-receptive cycle cases where we did not really see whether there was a higher number of FOXP3 positive cells or FOXP3 positive regulatory T cell percentage in any of the groups. So ERA assay would not really tell us the significance of immune cells that are contributing to successful or failed pregnancy. So let me take you through the conclusions. At this point, just with a limited number of samples, we are not able to predict conception based on phenotypic functional changes in pelvic NK cells in this time. As I showed you, pregnancy increases pelvic NK cells and regulatory T cells. Pregnancy and pregnancy serum lowers cytotoxic potential of pelvic NK cells. SCG in pregnancy serum plays a role in decreasing pelvic NK cell cytotoxicity. Infertile patients who do not conceive have similar endometrial NK cells, and a trend toward higher CD16 positive NK cells in this cycle prior to their frozen embryo transfer compared to infertile patients who do conceive. So, just to continue, infertile patients who conceive have higher numbers of endometrial Tregs when we characterize their endometrial tissue in the cycle prior to their frozen embryo transfer compared to infertile patients who do not conceive. This may be used for predictive testing prior to frozen embryo transfer. We did not find any differences in endometrial NK and Treg density in natural versus program cycle, or in, I did not show that data, but or in non-receptive versus receptive endometrium ERA cycle. And I did not show you the data, but we have the results now that TGF-beta converts pelvic blood NK cells into uterine NK cell-like cells by virtue of expressing CD9 and CD49A and by producing angiogenic factors. So I'll stop right here, and I would like to acknowledge the people who have done the work. As I mentioned before, Dr. Maytel, Soberner, Tattle, and Dr. Hoover were the main people who are now faculty members in REI division here, and Paula Kruger. These are the three people who really did the most of the work. with the help from other people in the lab. We have clinical core staff that collect the samples in the clinic, and wonderful collaborators who have helped us along the way, not only in this project but many other projects. And this work won't be possible without the funding from different sources for this work. So thank you very much. I wish I was there in person. I could take your questions, but have a good evening. Bye now.
感谢 Sharma 教授给我们分享了他们在那个做了一个外周血的 NK-CD56 和 CD16 那个NK 细胞和 Treg 细胞对于子宫内膜容受性以及决定妊娠是否能够成功的这些一些临床的一些探索。那么他这个研究其实对于临床医生来说还是蛮有意义的。那么对于 IVF 的病人，什么时候能够移植，什么时候能够找到最佳的一个种植窗，他都是做了一些探索性的研究。那么可惜就是没有提问的时间了。那么后面有请下一位讲者刘小同学。
我插一句，因为这个研究呢，一直是Sharma他长期做的工作。他因为担任这个美国生殖免疫协会的主席，包括杂志主编很久。其实呢，他是最后总结还是不能预测，但是他做了有一些趋势，比如说 CD16 阳性的细胞高了以后，它可能会影响种植，但是它没有更多的证据，所以我们要继续研究。因为 NK 细胞本身怀孕之后会增加很多，退活里面大概70%的都是 NK 细胞，但是这些细胞到底是什么作用？这个高了低了，对这个种植或对将来的流产到底是发生什么作用？现在没有人搞清楚，所以它没办法找一个预测的指标。所以现在包括同种免疫的流产也不推荐这些细胞，尤其外周血去检测是没办法预测的，所以还继续的研究。那么顺便说一个事情，就是最近呢，我们跟他合作，在中国要搞一个这个中国临床生殖免疫学的特刊，就是在中国搞生殖免疫的临床研究的文章可以推荐给我。我们呢，在就是美国生殖免疫杂志专门为中国的研究人员做一个增刊，叫中国特刊，都是临床研究的。所以大家如果有兴趣，有的稿件可以发给我，我会推荐给杂志。那么截止投稿日期应该是明年6月份之前。我们初步定的是6月份，就6月30号之前。大家如果有想法，可以把题目先给我，文章呢可以，那么最后整理再发给我。那么我呢担任中国的编辑之一，所以呢大家有兴趣可以推荐给我，好吧？